Assalamualaikum, my name is Nur Amina Khadija Ramli, my matrix is 73073 from group 183. Today our group will be presenting about accelerating drug design through evolutionary neural networks in molecular modeling. For introductions, drug discovery requires exploring a large and complex chemical space to identify molecules that bind specifically to biological targets. Traditional methods such as high screening throughput and quantitative structure activity relationship models are limited and cannot effectively cover this space or capture complex molecular interactions. Deep learning approaches improve modeling capability but suffer from bad behavior, local minima, and poor generalization to new compounds. Designing an optimal neural network architecture is also computationally expensive and relies heavily on trial and error. Evolutionary neural networks are proposed to overcome this issue by combining evolutionary algorithm and neural networks with neural networks enabling global optimization, adaptive architecture evolution, and improved performance in modern drug discovery. For problem background, we have 6. The first one is chemical space is vast and complex, making efficient exploration is difficult. For the second one, traditional high-throughput screening and quantitative structure activity model methods have limited coverage and accuracy. For the third one, deep learning model suffers from local minima and poor generalization. The fourth one, neural network architectures require manual trial and error tuning. The fifth one, protein ligand scoring lacks a balance between accuracy and efficiency. The last one, molecular representation struggles to capture complex structure information. For problem solutions, the problem solution that we have discovered is a evolutionary neural network. Evolutionary neural network is a hybrid intelligence system combining evolutionary algorithm and neural networks. Evolutionary neural network is use global evolutionary search instead of gradient-based optimization. It maintains a population of neural networks evolved over generations. It also applies selections, crossover, and mutations to optimize weight and structures. It effectively avoids local optima suitable for rare molecular landscapes. The last one is it produces model better adapted for molecular prediction tasks. For problem solutions, key capabilities and advantage we have 4. The first one is automated architecture evolution using NETs. The second one is support the novel drugs with design with GN and VAEs. The third one is it enables near quantum accuracy via transferable potentials, and the last one is they provide a robust, scalable, and automated framework for modern drug discovery. For comparison between the three models, we have provided a table. To summarize, traditional QSAR is simple, fast, and interpretable but limited to linear patterns. Deep neural networks can handle complex nonlinear data, though they require large datasets and act as black box. For evolutionary neural networks, it combines global search with adaptive structure, making them more suitable for complex and exploratory drug design tasks.
Hi, my name is Atira. S72877 will present about methodology. For the first methodology is algorithmic framework and hybrid architecture. For the methodology, this study proposed a hybrid intelligent framework known as Evolutionary Graph Neural Network or EVO-GNN. Unlike conventional deep learning models that rely on fixed architectures and gradient-based optimization, our approach uses evolutionary algorithm to evolve both the network structure and parameters dynamically. We begin with a population of heterogeneous neural networks, each representing a candidate solution for predicting drug target interactions. Through evolutionary operations such as selection, crossover, and mutation, the system explores a wide range of architectures while avoiding premature convergence to local minima. For the second methodology, which is molecular representation and graph processing. To represent molecular structures accurately, molecules are modeled as graphs, where atoms are treated as nodes and chemical bond as edges. Each atom and bond is encoded with relevant psychochemical features. These graphs are processed using a message passing neural network, allowing the model to learn local chemical environments in early layers and global molecular topology in deeper layers. This representation preserves structural information that traditional fingerprints often lose. For the third methodology, evolutionary optimization and intelligent integration. Optimization in this framework is performed using a evolutionary approach rather than backpropagation. All network weights and topology are treated as genome and optimized through genetic operators to protect structural innovation. A speciation mechanism inspired by need is applied. This allows complex and promising architecture to evolve gradually without being limited too early. The next one is fitness function and multi-objectives evaluation. The evolutionary process is guided by a multi-objective fitness function. Prediction accuracy evaluated using RMS is on binding affinity values. At the same time, its drug likeness and synthetic feasibility are enforced using quantitative estimate of drug likeness and synthetic accessibility score. This ensures that the evolved models prioritize not only accuracy but also chemical realisms. For the fifth one, data preprocessing and swarm intelligence integration. Before training, all molecular data undergo standardization using RDKit to remove noise and inconsistency. Particle swarm optimization is then applied for feature selection, reducing dimensionality and improving convergence speed. To ensure realistic evaluation, scaffold-based weighting is used, which tests the model ability to generalize to novel chemical structures. And the last one, justification of the hybrid methodology. Is overall, this hybrid methodology is well suited for drug discovery. Evolutionary learning address data scarcity and RAG optimization landscape, while graph neural networks capture complex molecular structures. Together, they form a robust end-to-end framework that overcome the limitation of traditional machine learning and standard deep learning approaches.
Moving on to our results and discussion, our quantitative performance analysis reveals that the ENN shows the highest prediction accuracy across GPCRs, kinases, and ion channels. Quantitatively, the ENN achieved a global RMSE of 0.42 and a R-square of 0.89, outperforming DNN, random forest, and SVM. By utilizing a scaffold-based methodology, the ENN demonstrates strong generalization and effectively capture active cliffs missed by traditional models. This precision allows us to reliably distinguish high-potency binders from low-potency compounds. Next, moving to our result regarding architectural evolution and topological insights. Our ENN architectures adapt automatically to target complexity using neural evolution of augmenting topologies. We found that kinases targets evolve deep, narrow networks for iterative refinement. In stark contrast, ion channels targets evolve wide, shallow architectures. This architectural plasticity confirms there is no one-size-fits-all architecture in drug discovery. Finally, integrating swarm intelligence for feature selection enables 40% faster convergence, representing a robust hybrid meta-heuristic framework. So next is our results and discussion on multi-objective ADMET optimization. This study pushes beyond single objective potency to identify compounds that are truly safe and deliverable. The ENN framework optimizes potency, drug likeness, and safety simultaneously. Our generated molecule achieve a high QED score of 0.82 with feasible synthetic accessibility, significantly outperforming traditional models. For safety, the system filters high-risk compounds with 92% accuracy in HERG risk detection. By penalizing toxicants, we produce chemically stable and orally viable drug candidates. Ultimately, this holistic optimization directly lowers attrition rates in late-stage drug development, tackling the industry's central economic bottleneck. So next is the benefits and advantages of the hybrid system. First, superior generalization and accuracy. The evolutionary neural network achieved a striking reduction in prediction error on unseen scaffolds, providing prediction with strong confidence. Second, automated architecture engineering. The system independently discovered the most suitable topology, meaning expert human intervention become largely unnecessary and manual tuning disappear entirely. Third, cost efficiency in screening. The false positive rate drops to only 3%, which reduces screen costs and ensures resources concentrate on compounds with the greatest probability of success. Finally, enhanced safety profile via ADMET array optimization. Built-in safety by design ensures toxic or insoluble compounds undergo proactive filtering in silico, reducing risk throughout the development pipeline. So for economic and strategy impacts, our result actively reinforce the fail early, fail cheap drug discovery strategy. By reducing the false positive rate from 15% to only 3%, we significantly reduce wasted synthesis and assay costs that traditionally stall development. The precision enables a fundamental shift towards autonomous drug design. It provides the intelligent necessities to overcome Eroom's goal. The observation that drug discovery is becoming slower and more expensive over time. Finally, this framework facilitates drug repurposing and democratizes discovery for smaller biotech firms, allowing computational power to replace the need for massive physical compound libraries. So in conclusion, the ENN framework combines evolutionary algorithms with neural network for efficient exploration of complex molecular space. Its global search capabilities overcome local minima, leading to more reliable and generalizable prediction. Automated architecture design reduces manual efforts, prevents overfitting, and adapts to diverse drug targets. By integrating graph neural networks and swarm intelligence, we more accurately represent molecular structure and learn from diverse data. Multi-objective optimization of ADMET properties ensure potency, safety, and drug likeness simultaneously. This holistic approach supports the fail early, fail cheap paradigm by reducing false positive and screening costs. Ultimately, ENN stands as a scalable, cost-efficient solution, reducing late-stage failures and enabling faster drug discovery, optimization, and repurposing. With that, thank you.